Good afternoon, everyone, and welcome to today's edition of Black Health Trust. We are so happy that you chose to spend some time with us, and I am always excited and eager about our shows, but I'm even more so today because I have really been looking forward to having our featured guest, Dr. James Hildreth, who is probably the most renowned virologist in this country, and from whom I have been wanting to have words of wisdom shared regarding COVID and anything else that we need to be worried about. So without further ado, I'll turn it over to our founder, Dr. Randall Maxey.
Dr. Maxey is on mute.
We lost.
I think I'm off mute now. Well, thank you very much, Dr. Barbara Neighbors-Stevens, who's an alumni faculty member of the Medical Center in Nashville, Tennessee. And we welcome you, and Dr. Walks, my associate from the East Coast, and Dr. Hines and others. We do, in fact, have a major speaker with us today. And I was trying to summarize his bio, and it still took me 100 slides to condense his bio. But very briefly, Dr. James E.K. Hildreth, MD, PhD, is the 125th president and chief executive officer of Meharry Medical College, the nation's largest private, independent, historically Black academic health sciences center. Dr. Hildreth obtained his BA in chemistry from Harvard University. He was the first African-American Rhodes Scholar from Arkansas. That means he likes goulash. He obtained a PhD in immunology from Oxford University, where his studies focused on the biology of virus-specific cytotoxic T cells. Dr. Hildreth also became an expert in monoclonal antibody technology and cell adhesion molecules. He also obtained his MD degree from Johns Hopkins University, where he also did a postdoctoral fellowship in pharmacology. In 2005, he became director of the Center for AIDS Health Disparities Research at Meharry Medical College. And in 2011, he became the dean of the College of Biological Sciences at UC Davis, and his first African-American dean and tenured professor in both cellular and molecular biology, as well as internal medicine. Dr. Hildreth's research in immunology and virology with a focus on HIV has resulted in more than 130 publications in top journals and 11 patents, and a technology that was developed by him was licensed by Genentech as the basis for the FDA-approved drug called Raptiva. Dr. Hildreth has received numerous awards for his research and his commitment to diversity in medicine. His recognition include honorary degrees. I understand he also has an SC, a DSC degree, which I believe only Einstein, Richard Williams, and a few other people have. And he was elected to the National Academy of Medicine. His leadership in the COVID-19 pandemic response saw him recognized as one of the top 50 most influential clinical leaders. Dr. Hildreth has been appointed to 2 FDA advisory committees, and he'll speak about those later. He was also, in February of 2021, appointed by President Biden to the COVID-19 Health Equity Task Force. He also currently serves on the advisory council of the NIH director as chair of that group. And I'm also predicting that he is going to be a strong candidate to replace Francis Collins as chair and Director of the National Institutes of Health. Dr. Hilders, we welcome you to the Black Health Trust, and we appreciate you agreeing to join us today. Our audience is primarily lay audience, but we have a number of physicians and that join us as we stream live on Facebook and YouTube and other things, and we appreciate you being with us. Thank you so much. The floor is yours, sir.
Thank you, Dr. Maxey, and I'm pleased to be able to share some thoughts with you today. What I thought I might do is to give a presentation that will answer most questions that people have asked me over the last 2.5 years. And then maybe I could take some questions to clarify some points that if I don't make them as well as I'd like to. So I'm gonna try to share my screen, and I hope you all can see that.
Yes, sir.
I start all of my talks actually by reminding people of the rich legacy and mission of the institution that I lead that's been around since 1876. As you heard, we're the largest private, independent, primarily Black academic health science center in the country. Our mission is to provide opportunities both for training and for healthcare for those who otherwise wouldn't have it. And I'm really proud to hold up that legacy and be a chief cheerleader for this amazing institution. Yeah, if I can get the slides to advance, that would be nice. So what I'm going to do today is give an overview of COVID-19 and talk about 3 things really. Very briefly, the virus itself, the disease it causes, and the vaccines that we are fortunate to have to protect ourselves. COVID-19 is caused by a virus called SARS-CoV-2 or severe acute respiratory syndrome coronavirus 2. There are 7 viruses in the human coronavirus family. The ones here on the left have been around since the 60s. They cause a common cold, and I venture to say everyone listening to me today has probably been infected by one of these 4 sometime during their lifetime. The 3 viruses on the left are the ones that have caused pandemics: SARS-CoV in 2003, MERS-CoV in 2012. And these viruses are examples of what are called zoonotic transmissions. When viruses jump from one species into another, that's referred to as a zoonosis, and these are examples of zoonotic transmission. There are more than 30, actually more than 40 known coronaviruses. Many of them are endemic to bats. And when bats interact with other animals, privets or camels, the viruses can infect those intermediate animals. And then those animals pass the virus on to us. So they're intermediate hosts. And these are called vectors. So privets were the vector for SARS-CoV. Camels were the vectors for MERS-CoV. And unless you came into contact with one of these animals, you did not get infected by these viruses. And that's why the SARS pandemic in 2003 and the MERS pandemic in 2012 were not nearly as expansive as the pandemic we're currently dealing with. The difference is this: these viruses are probably still endemic, the ones we're dealing with, endemic to bats. We don't know what the intermediate host was, but we think there was one. Many people think it might be these armadillo-like animals that you see here. Whatever the intermediate host was, humans interacted with it. The virus infected those humans. It quickly adopted for human-to-human transmission. So humans became the vector, and we are spreading the virus to each other. And that's why the COVID-19 pandemic is so much more problematic than either MERS or SARS, because we humans are spreading the virus to each other rather than having to have an intermediate host. The other thing that's really fundamental about this virus that must be understood, and it took the CDC way too long to acknowledge this, SARS-CoV-2 is a truly airborne pathogen. What that means is the particles that it rides on are so small, you don't have to cough or sneeze or yell or sing or do anything else. Having a normal conversation with another person for more than about 10 minutes standing in a conversation with that person is sufficient to transmit the virus. You don't have to work at it. You can simply have a conversation and be exposed. And that is why masks are so very important, and why masks became a political football, I do not understand. The reason why we use masks is very simple. In this cartoon, the woman at the top is asymptomatic. She's infected. She does not know that she's capable of transmitting the virus, but she's doing so. And these two ladies having a conversation with her, one is wearing a mask, as she should, the other not doing so. She is maximally exposed, she is minimally exposed. The real magic happens when both the person speaking and the person listening to the speaker are both wearing a mask. The amount of virus that's emitted into the air is drastically reduced, and the likelihood of getting that small amount of virus is reduced even further when both parties are wearing a mask. And that's why masks are so very important in stopping the transmission of airborne viruses. Viruses are incomplete life forms. They cannot replicate unless they get inside of our cells by definition. They do so by having an attachment protein. In this case, we've all learned about the spike protein of coronaviruses. The virus spike protein binds to a receptor on the surface of the cell. The two membranes between the virus and the cell become continuous. The genetic material of the virus goes inside of the cell, and then we have an infection. So until and unless the virus can bind to a receptor and fuse to a cell, infection cannot happen. But this is fundamental to understanding how membrane-bound viruses, like coronaviruses, get into our system. So viruses have specific receptors. For example, HIV, which is well known to all of us, binds to and infects cells in our immune system. And that is why it destroys our immune system, and that's what causes AIDS. What's really remarkable about SARS-CoV-2 is that its receptor is expressed throughout the body. It's found in blood vessels in the brain. It's found in the lungs, of course. It's found in the heart, the intestines, the kidneys, the testes. This receptor, because of its function in our body, is very widely expressed. What that means is that it's possible, and now we know it's actually true, that the virus is infecting multiple organ systems in our body, not just the lungs, but other organs as well. The other thing to know about the virus, which I think is really important, is it first starts to replicate in our upper respiratory tract, that is our nose and the back of our throat. It makes its way down into our lungs, and the infection of the virus of our cells and destruction of those cells is important. But the other thing that's equally important is that the immune response to the virus does damage on its own right. And between the virus destroying our cells and the immune response to the virus, we get this severe disease in our lungs. But the real point I want to make is there is sufficient virus replicating in the upper respiratory tract that we can actually transmit the virus without even knowing we're doing so. And this is a pre-symptomatic or asymptomatic transmission. And that's very important because there's a lot of virus that's replicating in our upper respiratory tract. And that's why people who don't know they're infected can transmit the virus. So as of about a month ago, less than a month ago, we know that more than 6 million people have been infected with SARS-CoV-2, and more than 6 million people have died. The truth of the matter is that for every person diagnosed with a PCR test with COVID-19, we believe there are at least 2 people who've been infected who are not aware that they have. So this 600 million people being infected could well be more than 1 billion people infected. Here in the United States, officially, we have about 80 million people have been infected with the virus. 950,000 people have died. But as I said before, for every person diagnosed with a PCR test, the evidence suggests that maybe as many as 2 persons have been infected without being aware that they've been infected, or they didn't come to our medical attention. So this 80 million could well be 200 million people in the United States who've been infected by this virus. And I believe that this prediction has already been reached of 100 million people infected and 1 million people died. So as you all know, this started in China in late December of 2019, early in January of 2020. The Chinese clinicians and scientists made a very interesting observation, even back then, early on in the pandemic, that 7 out of 10 people in China who died of COVID-19 had an underlying condition, a comorbid condition. High blood pressure, diabetes, heart disease, asthma, a stroke, congestive heart failure, and smoking was another major factor, especially for men, in terms of who died from COVID-19. This was consistent across the whole country. that the people who were dying of COVID-19, most of them, in fact, had an underlying condition. So it was predicted and predictable that when COVID-19 made it to the United States, that brown and black people were going to have a serious problem with this, because as you can see, African Americans and Hispanics have a much higher rate of all of these chronic conditions than white people do. And therefore, it was predicted and predictable that we would have a disproportionate burden of COVID-19 disease, because we have half of us have heart disease, a third of us or more have high blood pressure, many of us are obese, we have a lot more cancer than others do, and diabetes is rampant in our populations. And sure enough, these are data that I chose from midsummer of 2020 from 4 states, and they represent Louisiana's New Orleans, primarily Michigan, Detroit, primarily Illinois, primarily Chicago. And as you can see from these data, at one point in the pandemic, the rate of deaths per 100,000 people from COVID-19, for example, in Detroit, was almost 10 to 1. 10 people who were Black died for every 1 white person who did. This was consistent throughout the whole country, with very few exceptions. As a matter of fact, when the CDC compiled the data from a number of months of observations, it became obvious that American Indians, African Americans, and Hispanics were many times more likely to be hospitalized and to die of COVID-19 than the majority of white population. And this is something that has still been consistent even as I sit and speak with you. Now, children, thankfully, were not beset with a burden of COVID-19 nearly as much as adults were, but the vast majority of all the children who suffered from COVID-19, and certainly the mass majority of those who died, had an underlying condition, which meant the minority children were generally at much higher risk of dying of COVID-19 and getting severe disease, just like we saw in adults. So the best hope we had for dealing with a global epidemic or pandemic was to derive vaccines for this virus. And so, as I shared with you before, the spike protein of SARS-CoV-2 enables it to go into our cells by binding to angiotensin converting enzyme number 2. This is the receptor for this virus. The strategy for vaccines was to elicit antibodies, these large proteins, that would bind to the spike protein and literally prevent the spike protein from binding to its receptor. These are called neutralizing antibodies. So the purpose or the foundational principle of the vaccines in this case, let's make antibodies that can bind to the spike protein and keep the virus from infecting cells. And these would be neutralizing antibodies. I do want to make the point, so many people seem to be confused about this. Vaccines do not create a force field around us to keep viruses from getting into our system. Vaccines will program our immune systems to rapidly respond to the virus and eliminate it. So so-called breakthrough infections does not mean that vaccines are not working. They're doing exactly what we thought they would do, which is when a person gets exposed to the virus, either no infection would happen at all, or if an infection did happen, it will be quickly dealt with by the immune system. And those individuals will be either asymptomatic or mildly symptomatic when they've been fully vaccinated. But the point is, vaccines do not create a force field, keep the viruses out. They fight the virus once the virus does get into your system. Here in the United States, there were 3 platforms chosen for vaccine production. I just want to briefly talk about those because that also was the basis for some confusion. The oldest technology known to us for making a vaccine is to do one of 2 things. We can take the virus itself and inactivate it with heat or chemicals and use that as a vaccine, or we can take the spike protein itself that's responsible for binding to cells and use that as a vaccine. We introduce that into the body, the immune system recognizes it as foreign, and we make antibodies against it. Or there's another old technology. This is the oldest technology for making vaccines, either using the virus or part of the virus. The other technology that's been around for a long time is to take another common cold virus called adenovirus. We inactivate it. We insert the gene for the spike protein of SARS-CoV-2 into this other virus. This virus then becomes a carrier for a gene for SARS-CoV-2, in this case, the spike protein gene. The DNA gets expressed in our bodies. We make the spike protein, we then make antibodies to it. The technology that got most of the attention, and I guess rightly so, is mRNA technology, messenger RNA technology. So messenger RNA, and I'm going to talk about this later, they're like barcodes that tell the synthetic machinery in our cells how to make proteins or which proteins to make. So in this case, we have a messenger RNA that codes for the spike protein of SARS-CoV-2. We put that into a lipid capsule. We inject that into muscle cells. The mRNA causes the protein to be made. We make antibodies to it. So all three of these platforms have the same goal of generating the spike protein in our system so that our immune system can respond to it and make antibodies. Many people objected to mRNA technology because they thought it was really new and untested and didn't want to have anything to do with it. The truth is, mRNAs were discovered in 1964. They've been used in medicine for 2 decades at least. They've been studied in animals since the time they were discovered back in the 60s. So while mRNA technology is new to vaccines, it's certainly not new to medicine. The other thing I was asked about is what is in the vaccines, and I'll just give you one example. People thought that there are going to be all kinds of crazy things in vaccines. Here's, in that vial you go to, when you go to the clinic to get your vaccine injection, that clear liquid that's being injected into your arm, this is what's in there. The mRNA to encode the spike protein. There's a mixture of lipids, including some cholesterol. There are some buffer salts that are inorganic and totally safe. There's a little bit of sugar and some water. And occasionally, for example, with Johnson & Johnson, there's a tiny bit of hydrochloric acid to adjust the pH. There are lipids that form the shell around the mRNA. And because of the instability of these mRNA lipid complexes, they have to be stored frozen at very low temperatures. The point I want to make is the vaccines that are currently being used do not contain microchips. If you're worried about being traced or followed because of the vaccine, put your cell phone down. That's where the chip is that follows you around. The vaccine is not going to do that. There are no preservatives. There are no heavy metals. There are no cellular products, no animal products. This is a sum total of what's in the vial when you go to get injected with your vaccine. What's really remarkable to me is less than 3 years into this challenge, we now have almost more than 12 billion shots have been given into arms from the 23 approved vaccines around the world. And as last time I checked, almost 5 billion individuals, 2/3 of the world's population, has now been vaccinated with at least one shot of one of these vaccines, which is truly remarkable. Here in the United States, between Pfizer, Moderna, Janssen, 610 million shots have been given in the primary series. If you add into the booster shots that are now being given to people, this is going to be north of 700 million shots that have been given here in the United States. So anyone who thinks they're going to be a guinea pig now in getting one of these vaccines, that's really a foolish thought, given that 12 billion shots and more than 5 billion people around the world have now been injected. No arms have fallen off, as far as I know, and other kinds of horrible things that people thought would happen have not happened. The data is very clear that the vaccines are extremely safe. And unfortunately, here in the United States, only about 68% of us are fully vaccinated, which is really remarkable to me, which means there are tens of millions of people out there who have still not been vaccinated. And I'm going to talk about why that's a problem a little bit later. The other thing I was asked over and over again is, why should we trust the vaccines? Because they were produced so quickly. And that is true. The genome for this virus was published by Chinese scientists on January the 10th. We had a vaccine before the FDA by late November for review by the FDA. So how is it possible that we did this in 10 months? Vaccines have taken as long as 100 years for tuberculosis and as fast as 4 years for Ebola. So the fastest vaccine we know of took 4 years. How can we do this so quickly? There are 3 things that made it possible to develop vaccines in 10 months, whereas before it took many years to do so. The first is technology. The technologies brought to bear for vaccine production in 2020 are truly astounding. They include molecular genetics, big data, molecular modeling, high throughput systems, and lots of others. So technology was critical. The other thing is that your tax dollars and my tax dollars in the billions were given to the drug companies so that rather than doing steps sequentially, they did the steps in parallel. Vaccine production and drug development is typically an iterative process. You do one step, you follow that by another step, and these steps can take months, sometimes years. But the drug companies were able to do them in parallel because of all the resources provided to them. And lastly, we've been trying to produce an HIV vaccine since 1984. We don't have an HIV vaccine. What we do have is a worldwide infrastructure for vaccine development that is truly astounding, consisting of scientists like myself, infrastructure, technologies, protocols, methodologies. And in March of 2020, essentially everyone working on HIV vaccines paused their work and turned their attention to COVID-19. So this vast infrastructure for vaccine development shifted its focus from one virus to another. So there are three major reasons we are able to do this in 10 months: technology, parallel processes, and an amazing infrastructure that was pre-existing. The other question I've been asked was, why should Black folks accept something that's been produced by medical research and medical technology, given a long history we've had of being the subject of, and not a very good subject of, medical research? It's true. We have the Tuskegee experiment we all know about. We have the involuntary sterilization of Black and brown women. In fact, I tell people, I can find atrocities visited upon Black bodies all the way back to 1619. So I accept that we have a basis for being mistrusting of medical research. But this time is very different. At every stage along the way, people of color have been involved. In terms of developing the vaccine technology itself, a brilliant, amazing young scientist named Kizzmekia Corbett at NIH played a pivotal role in getting mRNA technology to the point that it's ready for vaccines. She's now a professor at Harvard Medical School. When the vaccines are ready for human trials, there's something called a data safety monitoring board. These are independent citizens, scientists, physicians, patient advocates who get to monitor the progress of the trial. They get to see the data along the way. In fact, the Data Safety Monitoring Board, they're the only ones who know who gets the vaccine and who gets the placebo. The participants don't know, the physicians in the trial don't know. The Data Safety Monitoring Board members, they are the only ones who can tell who is a vaccine recipient and who is a placebo recipient. And they get, if they choose to, they can stop a trial by being concerned about what's happening to the participants. And every one of the data safety monitoring boards, there have been Black and brown folks, and my good friend Lisa Cooper, who's a professor at Johns Hopkins School of Medicine, served on a couple of these DSMBs. And then when the trials are over and the data makes its way to the FDA convenes a panel of independent scientists on something called the Vaccines and Related Biological Products Advisory Committee to review the data and make recommendations for approval and non-approval. Myself and my good friend and colleague, Dr. Avila Fuller from University of Michigan, we're the two African Americans who sit on the FDA VRBPAC. So my point is that at every step of vaccine development, there have been people of color sitting at the table. So we've been part of the decision-making process, the scientific process, the review process. So this has been very different. So all of us can be confident that people with our best interests in mind have been a part of this process from the beginning. Let me just talk a bit about why boosters are necessary. What we know from lots of work is that when people become vaccinated with this vaccine, with the vaccines we have, because we're all naive to this virus, naive to the vaccine, our immune system does not have a sustained response to it. So what this data simply shows you is these are neutralizing titers of antibodies in our system. And this is a log scale. So each one of these increments, 1, 2, 3, represents a 10-fold increase in antibody levels. The point I want to make is that after 6 to 8 months, as you can see, the neutralizing antibody titers have dropped off by at least 1 order of magnitude, and in some cases, 2 orders of magnitude. They're 100-fold lower than they were when the primary series of vaccines were given. And this is why we knew that boosters would be necessary to keep the levels of neutralizing antibodies sustained. To give an example of what that looks like, here's some data for the Moderna vaccine showing you what happens when you get a booster shot. So here is sort of day 1 after you finish your series of infections. And here's what happens when you get your booster shot. That we see that there is a 10-fold, in some cases almost 100-fold increase in those neutralizing antibody titers, right? So you can actually see that the boosters do exactly what we thought they would do in terms of re-elevating or sustaining the antibody levels you need to neutralize the virus. So the data from Israel are very clear. I won't spend any time on this, but Israel was out in front in doing research on boosters and sustaining antibody levels. Basically, I just want to show you that when you look at persons who got 2 doses of the vaccine versus 2 doses plus a boost or 3 doses, if you look at the cumulative events of hospitalization, severe COVID-19 and deaths, when there were 3 shots, the rate of all of these things was clearly much less than if you only had the primary series. So the data from Israel were very clear that boosters, especially in older adults, did what we thought they would do, which is to drastically reduce hospitalizations, severe COVID-19 and deaths. So let me say a few words about variants, and then I'll stop and answer questions. All RNA viruses, like COVID-19 virus, SARS-CoV-2, they mutate. That is to say, the sequence of their genome changes as they replicate in cells. None of them do so like HIV. HIV mutates faster than any virus we've ever known, but all RNA viruses mutate. The mutations, as I'm going to show you, change what I call epitopes. Epitopes are what our immune system sees and responds to. So when viruses mutate and change their epitopes, it makes it more challenging for our immune system to keep up. And SARS-CoV-2, by infecting hundreds of millions of people, have had ample opportunities to mutate and therefore for variants to arise. And as long as there are large groups of people who are not vaccinated who can replicate the virus, there's always a chance that a variant could emerge. And that is why we thought it was really important to get as many people vaccinated as quickly as possible to prevent variants from arising in the unvaccinated persons. So here's a little virology 101 to help you understand why this is so important. So as I shared with you, coronaviruses have to get inside of our cells to replicate. One of the very first things that happens is there's a protein called a virus polymerase that copies the RNA to make hundreds and hundreds of copies of the RNA so that new viruses can form. But this protein makes mistakes. And when it makes mistakes, those are mutations. And those mutations are what allows the virus to change its epitopes and become variants. And that's what happens here. That as this protein, as this RNA is being replicated, the protein that does so makes some mistakes in copying it, and we get mutations. So you can think of mRNA as a barcode. There are a series of barcodes that are strung together to tell the machinery in our cells which proteins to make, which amino acids to join together. And they're called codons, actually. And there are 21 amino acids that make up our proteins. Each one of them has its own barcode. And as you can see, some of them have more than one barcode. In fact, valine and alanine have 4 barcodes each. But the point is, when mRNA is made, we string together barcodes. In this case, there's a barcode for valine, one for alanine, glutamic acid, and glycine, because these are the barcodes that are part of the mRNA. So if a mutation occurs, glutamic acid is a negatively charged amino acid. Let's just say we change one barcode by changing one position. We change that G to an A, and that happens when we're copying the RNA of the virus. Now, instead of having a negatively charged amino acid, like glutamic acid, we have a positively charged amino acid in lysine. You may think that's a trivial change, but it's not. So when antibodies are made, there's strings of amino acids. Those amino acids fold on each other to form shapes. And sometimes those shapes are held together by salt bridges. As all of us know, opposite charges attract each other. So in this case, glutamic acid is binding to lysine to make a salt bridge, and that will confine the shape of this protein. And an antibody can bind to that shape. But if a mutation occurs, like I just showed you, and that negative charge goes away, the protein will be unfolded, the shape will change, and there's nothing for the antibody to bind to. The point is that when changes occur in the barcodes, antibodies cannot bind, and that is what variants represent. Variants are simply changes in the barcodes that change what the immune system sees and might disallow antibodies to bind. And that's why variants are so concerning. The good news is that the genome of SARS-CoV-2 is itself an mRNA. It's a positive strand RNA virus, which means that the genome itself is a glorified RNA. What that means for us is that if you make a vaccine that is also an mRNA, when the virus mutates to change its mRNA sequence, we can actually go back into our lab and make exactly the same changes in the mRNA for a booster shot. In other words, the mRNA vaccine can copy exactly what the virus did. So now the vaccine matches the virus. And that is what Pfizer and Moderna did to make their booster shots. They went back into the laboratory, identified the changes that have been made in Omicron in the RNA of the virus. And they made exactly the same RNA changes in their vaccine. And that is one of the nice things about mRNA technology. We can quickly match change for change what the virus does. We can do exactly the same thing in the vaccine. And that is what variants are. And that's what these booster vaccines are doing. They're simply allowing us to copy what the virus is doing. Now, it's going to be very challenging to do that for all the variants that occur, but for those variants that are variants of concern, this is a beautiful thing for us to be able to do, and it can be done very quickly. The one challenge we have is whether or not the FDA will make us test each and every booster vaccine as a new substance, because in fact, we've created a new chemical. This mRNA with these changed base pairs are not the same as the original. This is a unique chemical entity. So the FDA has to decide whether or not we'll require a full human trial to evaluate this new vaccine compared to the original one. But hopefully this explains what variants are, why they're caused, and what the booster vaccines are doing. So the good news is that the variants have the same biophysical properties as the original strains. Masks will control them just as well as they did the original strains of virus. Variants are subject to the same sanitizers and detergents as the original ones are. And thank goodness masks, physical distancing, and all the other things we've been doing for the original strains of virus that work just as well for the variants. And the data we have so far indicate that the EUA approved booster shots will do well against Omicron and the other variants. And the last point I want to make is, this point needs to be made as often as possible. The data continues to show that virtually every person who is now being hospitalized for COVID-19, and for those who are dying, they're unvaccinated. Vaccines work. They do not necessarily keep you from being infected, but they will keep you out of the hospital and they will keep you alive. So we need to make sure that those persons we know in our circles who are not vaccinated, we need to do everything we can to get them vaccinated, because it's the only hope of saving lives still, as I sit and speak with you here in October of 2022. With that, I'm going to thank you, and I'll be happy to answer questions.
Dr. Hildreth, thank you so much for your extremely beautiful and very cogent lecture on viruses. I think I understand almost everything now, but I do have an initial question. Last week, the CDC came out with what I consider a strange set of new guidances that have led some hospital entities to relax their use of masks. And my daughter's a nurse in a hospital in upstate New York, and she says now the patients don't have to wear masks, the visitors don't have to wear masks, nor do the medical staff and nursing staff. What's your comment on how that guidance is? And it's not necessarily related to what's going on in the community, it's related to the transmission rate, I heard.
Sure. So Dr. Maxey, I can just tell you here at Meharry Medical College, in our clinics and in the hospital, masks are still required. You can't, the same is true at Vanderbilt, and I was just at Johns Hopkins 2 weeks ago. They're still requiring masks of patients and medical staff. I think that the CDC has been off the mark, and I have great respect for my colleagues there, because there's some amazing scientists there. But some of the decisions that are being made, quite honestly, from my perspective, seem to be driven by politics more than anything else. And I would advise anyone who is going to be part of a large crowd where you know most people are not vaccinated, that you still protect yourself by wearing a mask. I've started flying again. I'm wearing my mask in airports and on airplanes. This is not over, okay? The pandemic is not over. As long as there are tens of millions of people out there who are not vaccinated, and as long as there's virus in circulation, it allows for some variants to occur that might be very challenging to all of us. So I'm just saying that from our perspective as medical professionals here at Meharry Medical College, in the clinics, on the wards, our students, residents, physicians, and patients are all required still to wear a mask. So I don't know what's happening in New York, but I can tell you here in Nashville, that has not changed.
And thank you. Dr. Neighbor Stevens, Dr. Walks, you have any comments? Is there anything in the chat?
Dr. Hilder, thank you so much for this very clear explanation of where we are now. As a pediatrician, I listened with a great deal of interest, trying to find key terms and pearls to share with parents, because as I'm sure, I know you know, not only is there vaccine hesitancy regarding COVID, but there's also a huge increase in vaccination hesitancy. Actually, I call it resistance, to the childhood vaccines that we've been given for years. Do you have any words of wisdom for those of us who interact with the public to try to quell their fears about vaccinations?
Well, it's something that we all should be really concerned about, because I'm sure everyone in the Brady Bunch boxes here have all been given childhood vaccines. And there are tens of millions of us, hundreds of millions of people around the world who were given vaccines as children. And most of us had no issues with it. So we know that those are safe. The reemergence of polio and measles are indications that this vaccine hesitancy and resistance might be starting to have some very untoward impact on our population. And we're really concerned about it. I haven't found the answers that would necessarily satisfy those folks who feel this way, except to say that now that we have millions of people in the United States, hundreds of millions, in fact, who've been vaccinated, including tens of millions of children, the safety, as far as I'm concerned, is in the numbers and in the evidence in front of our own eyes, right? And the fact that our children are more likely to be severely impacted by COVID-19 and to die from it means that we should be focused on this issue much more so than maybe other populations should be. I know that the indigenous people are really concerned about this because they have the same issue that we do. And we partner with a number of tribes to try to find the right message message and messengers in that population. And one thing I've learned in all of my efforts over the last 2.5 years, if we don't have trusted messengers, it really doesn't matter what the message is. It's going to fall on deaf ears. So my advice would be to try to identify those persons or persons who are trusted and respected by the people we're trying to reach, and have them be the message bearers for us. And that's what I've tried to do with faith leaders, with young people, and through the work I've been doing for the last 2.5 years. So I wish I had a better answer for you, but I do agree with you that it's one of the most pressing issues we have right now, is convincing parents of children to make sure their children get those vaccines that will keep them protected, not just from COVID-19, but all those things that we worry about that are reemerging as we sit and talk here today. But trusted messengers are vital to this fight, without any question whatsoever.
Wow,
Thank you.
Dr. Hildreth, I'm sitting here smiling because I just appreciate you. I appreciate the scholarship. I appreciate the clarity so much. I appreciate you going to my alma mater, UC Davis, and adding a little color. There was none when I was there. And I appreciate my nephew graduating from Meharry Medical College. He's now a resident a couple of years ago. So I'm sitting here smiling so much. I have a couple of questions, but let me go to the chat, because one of the questions in the chat is, and I'll just abbreviate it. The assumption is that since the spike protein can mutate, but the assumption is the virus body does not mutate, why not make a vaccine against the virus body so you wouldn't have to do all of the boosting when the spike changes? If you could just comment on that, please.
Sure. And I think the person who asked that question must be an immunologist, because there are two ways that our immune system fight viruses. Antibodies attack the virus particles themselves, but the cytotoxic T cells actually kill the infected cells that are kind of the virus factories. So antibodies eliminate the virus that's already been made. The cytotoxic T cells eliminate the cells that are making the virus. And in fact, if we had a whole virus vaccine, there are a number of other proteins that get made inside of our cells, and our cytotoxic T cells can see fragments of those proteins on the surface of those cells and kill them. So I worried from the very beginning, and I actually said this in the very first VRPAC meeting, isn't there a concern that by focusing on spike protein, we're not taking advantage of that huge other part of the immune system that could be destroying the infected cells? So we now have some evidence that T cells do play an important role. And I know for a fact that Sandoz Pharmaceuticals, Novavax, and a few other vaccine companies are doing exactly that, for exactly that reason. And those proteins tend to be more conserved than the spike protein, because they serve an unchanging function for the virus. So whoever asked the question is an excellent question. And some of the vaccine companies are now focused on doing exactly that. And one of the vaccines that was approved in China and some other places was the whole inactivated virus particle. You just take the virus particles, you grow them up into large numbers, you inactivate them with heat or chemicals, and you use that as a vaccine. In fact, the very first vaccine was in fact a whole particle vaccine that had been attenuated to not replicate. So it's an excellent strategy, one that I think we should have been using from the very beginning.
Thank you, Dr. Hildreth. There is one other comment in the chat, and then I'll keep my question and pass it over to Dr. Hines. There's a question in the chat requesting that you please discuss some situations in which the vaccine may not be appropriate to give to an individual.
Okay. There are a number of situations that I think would fall in that category. I've often been asked that if you have COVID-19, should you wait to get vaccinated, and why should you? When people become infected, they make antibodies to the virus. And those antibodies may not be protected, but they will bind to the proteins that the virus makes. And so the antibodies you make to the spike protein in response to infection, if you were to be vaccinated too soon, those antibodies would bind to the spike protein and through a negative feedback loop, prevent your immune system from responding. Most of the systems in the body work this way. For example, insulin is made by the pancreas. And when there's enough insulin made to get our sugar in balance, it gets turned off by a feedback loop. The same thing happens in the immune system. So if you get vaccinated while your antibody levels are high from being infected, the vaccine will work for you. And that is why we recommend that persons who've actually had COVID-19 wait 3 months or more before they get vaccinated to allow those antibodies levels to drop so the vaccine can work. Those individuals who might be on treatment for autoimmune diseases, such as multiple sclerosis, those drugs suppress the very cell types that make antibodies. So if you are on those treatments and you get vaccinated, the vaccine won't do you any good because that drug that's suppressing your B cells will suppress the B cells that would be responding to the vaccine. So you need to have a conversation with your physician about other alternative treatments for the ones you're getting to allow the vaccine to work, or should you get passive vaccination? By that I mean, would monoclonal antibodies be a better option for you in that case? So those are just two examples where you might think about getting something other than waiting to get the vaccination or getting passive immunization by getting either monoclonal antibodies or some other treatment. Those are just two examples. There are others. But those persons who are worried about that, having a conversation with your primary care physician would be the best advice I could offer.
Thank you, Dr. Hildreth. Dr. Hines, and then Dr. Maxey, Dr. Gibson had his hand up.
Okay, very good. Dr. Hines, how are you?
I'm great, thank you. How are you? My question is really around the concept of over-vaccination. So now that we've reached a space where we're talking about boosters periodically and flu season's coming, I've definitely heard a lot of concern around, how many vaccines how many shots is too many? What's going to happen if I have all of this in my system? I was just wondering what you might say to people who have those concerns.
It's a legitimate concern. There's something that we immunologists refer to as original antigenic sin, that if you get exposed to the same challenge to your immune system multiple times in a short period of time, your immune system becomes focused on that one thing, which means that if a variant comes along that's substantially different from that one thing, your immune system will be challenged to respond to it. And that's why I was advocating for heterologous boosters. For example, I got the Moderna vaccine as my primary series, but I got the Pfizer as my booster. And I'm going to get the Novavax vaccine as my fourth shot to avoid original antigenic sin and my immune system become focused on just one thing. Besides that, the data for the Novavax vaccine, which is a protein, show that the antibodies we make to the Novavax vaccine neutralizes all the variants that are out there at the moment. That is not true necessarily for the other vaccines. But there is a legitimate concern that too many vaccinations within too short a period of time can cause our immune system to be super focused on a narrow range of antigens, and that would be a bad thing. So I waited a whole year between getting my second shot and getting boosted for that reason. Okay, I did all the steps I needed to protect myself, masking, social distancing, all that stuff. But knowing what I know, I didn't want to get myself boosted too soon after the primary series, so I waited. And I would advise anyone to wait as long as you can between your primary series and getting boosted, and between your third shot and your fourth shot to avoid that. So that is a legitimate concern. And whoever asked the question is a really great question. So my advice would be to take the precautionary steps you need to take to protect yourselves, but wait as long as you can between shots. One of the things I've complained about, Dr. Hines, from the very beginning is not having what we call a correlate of immunity. In other words, is there a blood test we can take to tell us whether or not we're protected? It might be a certain antibody type at a certain titer. It might be a certain epitope we recognize that we know is on the virus. Just something that we could do to tell us that we're protected, to allow us to get a simple test to say, okay, you don't need a booster. Because the longer you can wait, the better. But this whole idea of getting a shot every 4 or 5 months makes me nervous because of that concept of original antigenic sin. So, and I do believe that there's some folks at NIH and WHO that are working to identify a correlative immunity. And once we have that, we'll have a better sense of when boosters will be necessary by having a simple test. So that's a great question. Thank you.
Thank you. Dr. Walsh, do you have your own question or?
Dr. Maxey, I did have my own question, but now I'm dumbfounded and flabbergasted. So, Dr. Hildreth, as someone who watches and listens to all of this, and Dr. Fauci and everybody else for years, and all, why hasn't anybody ever mentioned? See, and Dr. Maxey would knock people down to get to his next booster as soon as they were available. And now you're telling me that Dr. Maxey should have been in the back of the line to get his next booster. I'm a little, could you just clarify whether Dr. Maxey has been doing stuff wrong or I've been doing stuff wrong?
No, I'm not saying that people who are following the CDC recommendations are making a bad decision. What I am saying, though, is that strictly from an immunological point of view and understanding how the immune system works, to Dr. Hines' point, getting boosted too frequently may be an exceptionally bad idea. What we need is some way we can do a clinical test, a simple test to tell us whether or not a person is protected, right? But we got to make sure our protection matches. So one of our challenges is we don't have a correlative immunity, but more importantly, we have no way of predicting when a variant will arise that challenges the vaccines we have available to us. I just shared with you that mRNA vaccines give us a great way to quickly make a vaccine that matches a variant of concern. But we don't know that changing the sequence of an mRNA won't create a unique substance that might have an off-target effect. And do we have to test each mRNA vaccine as if it were a completely new substance? So the complexities here are kind of mind-boggling. And I don't expect an ordinary layperson to understand all these things that I'm saying. I do wish the CDC was more communicated more effectively about what is happening and why they're recommending things to people. Okay. I can't unknow what I know, right? I just can't do that. But until there's evidence that frequent boostings are causing this antigenic sin I refer to, I guess we'll just have to go with it. But generally speaking, because of the way the immune system works, the longer you can wait between your booster shots, the better off you are. Because you don't run the risk of your immune system become so focused on one epitope that it won't respond to the next thing you have to be boosted to. Does that make sense?
So, no, Dr. Hildreth, that is clear. Dr. Maxey, we have a few comments and questions in the chat. We have a couple of hands up, and I'm just trying to make sure we respect Dr. Hildreth's time. I don't know how much time we have with him today.
So as long as he can stay with us, but let me put up a question. First of all, I'm 200 years old. I'm specifically at risk, so I got everything I could get, Dr. Hildreth, when I got it. But as we move on to recognize some of our other questions from our respected advisors on the phone, I want to make sure that at some point you comment about post-acute syndrome, how it happens, why it happens, and what we should be concerned about and how we treat it. But I think, was Dr. Gibson next? And I saw Dr. Clark's hand. Dr. Walks?
Yes, Dr. Gibson put his hand down. I think his question was probably answered. And Dr. Clark has her hand up. And I think I saw Dr. Sherrod's hand up.
Actually, I didn't put it down, but that's good.
Go ahead, Dr. Gibson.
Well, no, I'm curious about the correlative immunity comments that Dr. Hildreth made, along with the messaging. And on top of that, I would love to see an approach that combined B cell protection along with mRNA protection. I'm intrigued by that approach. What say you, good doctor?
Well, as I said from the beginning, when they first asked me to serve on the VRPAC and I started to see the data from the drug companies, it became very clear they're focusing only on antibodies. And to make a vaccine quickly and evaluate it quickly, that might have been the right choice, because if an antibody neutralizes, we can measure that in the laboratory within a matter of a couple of hours. So it makes screening the vaccines easier, it makes everything easier. But just because it's easy doesn't mean it should be the only thing we're focused on. And since my work in immunology has been on how T cells respond to infected cells, I have some knowledge about how important that part of the immune system is. But I'm thankful now that there are some of the drug companies, from vaccine-focused companies, that are trying to make vaccines that both induce neutralizing antibodies and killer T cells. And that is what the flu vaccines is supposed to do, actually, is to elicit both antibodies and cytotoxic T cells. And I think that the correlate of immunity is really important, because we give people a vaccine, we measure their antibody levels, and we call it a day, but we don't know if those antibodies are effective in doing what we want them to do, which is neutralizing the virus. And there's some extrapolation that happens from studies in mice and studies in macaques or monkeys to what happens in humans. And I can just tell you there are horror stories about making assumptions about how mice respond to certain things and thinking that humans will respond the same way. That has led to some unfortunate results. So we need to have a way that you can go to your physician and a simple, quick test, point of care test can be done to tell us whether or not you are in fact good and protected against COVID-19. And that way we could actually, save some resources, because we don't have to boost millions of people. We can wait, save some time and some resources for other things. But more importantly, we can prevent this thing that we as immunologists worry about, getting our immune system so preoccupied with one thing, they can't respond to something else.
So is it your position, good doctor, that those whole vaccines, that whole vaccination approach can be as nimble as the current mRNA technology and quick?
There is some clear evidence from some studies now being done by folks at Stanford, Harvard, and some other places, that the response that is made to a whole virus versus just a spike protein results in cytotoxic T cells that can recognize and kill viruses across multiple different sequences, different strains, different variants. And I think that's the impetus to try to make that effort front and center. Because if we're going to have a universal vaccine for coronaviruses, it may well be that is based on non-spike proteins that mutates faster than the other parts of the virus. There's some things the virus has to do that if it mutated those proteins, it could not do those anymore. For example, the protein that copies the RNA to make more copies of itself, that protein tends to be very stable and doesn't change very much. If it turns out that cytotoxic T cells can kill based on that protein, well, there you go. That could be the basis of a universal coronavirus vaccine, because you would elicit T cells that would kill all the viruses.
Thank you so much.
Thank you.
So while we're getting Dr. Clark unmuted, I just want to clarify, Dr. Hildreth, someone put in the chat, should I be scared because I've gotten that 5th shot? And no. I did not hear you say that. I heard you say that people should take their shots as advised by the CDC. Is that clear?
Correct. Yes. And there's research going on right now to answer that question I posed, which is whether or not we are in danger of eliciting the antigenic sin focus of the immune system. And if that happens, believe me, I will let the whole world know.
Thank you.
Because I'm worried about that myself. And so the research is underway to answer the question.
Thank you. Dr. Clark?
Good. Thank you so very much for the talk. I really enjoyed the presentation. You really just broke it down, to where it's understandable. the question I kind of have is just like what you said. I was actually recommending people to actually have antibody testing done before they got the booster shot when it first came out. And then, of course, when the wave came out with this whole protocol, well, that just pretty much went out the window.
Yes.
So my concern has been, again, about the over vaccination of of our people. And on top of that, when I relate it to like the flu shot, okay, we get that once a year. So why are we having to get this, the fourth and the third, the fourth and the fifth, even before pretty much the year is out? So, but you said something that did make sense. If we are having variants to arise, when we change the vaccine and now we're including those variants, I can understand really moving towards trying to get boosters to kind of cover that.
Right.
I don't necessarily know if I agree with just getting the booster, just to boost your antibodies when we're not testing to see where your antibodies are in the first place.
Exactly.
So
I
guess
we're on the same page, Dr. Clark.
Yeah.
And as quiet as it might be kept, and I'm not advising this for anybody, there are 3 studies I'm aware of to show that if you are fully vaccinated, that has gotten 2 primary series, Moderna or Pfizer, and you happen to get infected, you get COVID-19, when serum from those persons have been studied, their antibody levels are even higher and with a neutralization capacity if they received a booster.
Mhm.
Okay, so it's almost as if the infection itself was a boost to the immune system. And if you look at the antibodies in those persons, they neutralize a whole range of variants, beta, delta, the whole range. It's but it actually kind of makes sense from an immunological point of view, that you're vaccinated and now you get exposed to the virus. There's this huge response. There's a tertiary response, so it's not surprising that the response is so robust. And I tell people, if you've been vaccinated and you happen to get COVID-19 and recover, you should consider yourself boosted and wait before you get your shot, because for all practical purposes, your infection has served as your boost. That makes sense. Right.
Oh, that is great. There's a doctor visiting from Puerto Rico, Dr. De Silva. Would you care to make a comment?
And then we can go to Dr. Sherrod, Dr. Maxey.
Yes. Do you see Dr. De Silva? Can you unmute him, or you want to get Dr. Sherrod first? Is okay.
I see Dr. De Silva is unmuted.
Okay. Hi, everyone. Dr. Da Silva from Puerto Rico. Thank you, Dr. Maxey, for this invitation, and thank you for this great presentation. This is a great opportunity to let you know that Puerto Rico has a great rate of vaccination compared with other states. Yes. And it has been something that it has been well worked from the very beginning, because of various reasons, including that we didn't politicize this issue. We involved the community. Really, the approach was done based on the communities, and also we are used to work with limited resources, but in these specific instances, the government treated us, Puerto Rico, the same as they treated the other states, and we really take advantage of that and make a good work with the resources that they allow us to use for our population. So Puerto Rico has a very good rate of vaccination, and that has allowed us to really control the pandemic locally. So, and this kind of information is the one that is used to orient our patients and and you really use the science as the only base to convince the people this is the way to control any pandemic. So thank you for the information. It has been a really very good information for all of us.
Thank you, Dr.
Thank you,
Silva.
Dr. Sherrod.
Oh, thank you so much. Thank you, Dr. Hildreth, for such an excellent presentation. And it was broken down to the point that even I could understand it. I'm honored to have the opportunity to see you so soon again. My question is concerning the original sin, I mean, the antigenic sin and the fallout from that. And is there data or are there data on what's happening as a result of the boosters and the possibility of that? Because there's a lot of concern in the community that the boosters and all of the vaccinations are actually increasing the rate of cancer. People are talking about, well, I'm seeing more breast cancer, I'm seeing more blah, blah, blah. So what is the impact on the ability of the immune system to protect the body from other, antigens? And then the last question, I agree with all of the testing or getting a test to test people before you give the booster. And my dentist asked me, well, why can't I be tested before I get a booster? So thank you so much though for this presentation, because it has clarified a lot of things for me and I'm sure for the general public.
Thank you, Dr. Sherrod. As I said earlier, there are investigators in a number of places who are taking serum or antibodies from vaccinated persons who've been primary series only, 1 boost or 2 boosts, and they're starting to look at whether or not their ability to respond to other things. For example, this is flu season or time to be getting your flu vaccine. So one of my colleagues at NIH is collecting samples from about 3,000 individuals who've been double vaccinated with COVID and flu. And they're going to be looking at whether or not the response to flu is attenuated based on the boosting for COVID-19. The preliminary data that they've gotten so far suggests that there's no impact. And one of the things that the pharmaceutical companies did in the trials was to do similar studies on a more limited basis. There are tens of thousands of people that took part in the trials, and there were subgroups in those trials who got vaccinated with other things. And they looked to see whether or not those vaccinations for flu and some other viruses would be impacted by the COVID-19 vaccine. And the data we have so far, and it's accumulating rapidly, suggests that getting vaccinated with COVID-19 does not affect your ability to respond to other vaccines. But as you said before, if we're going to get 3 or 4 vaccines for COVID-19 in a short period of time, there are some of us, and I'm one of them, who worries about what we've known about for decades, and it's called antigenic sin, where you get the immune system so focused on one thing, it can't respond to another. And there's a protein marker we can look for on B cells that tell us that this is happening. And a lot of my colleagues are looking to see whether or not that expression is happening. So we'll have some answers here pretty soon to help guide us through this. And someone said, is there an antibody test that can be done? And the answer is yes. There are tests that look just for antibodies to spike, which tells you that you've been vaccinated. And there are antibody tests that looks for antibodies to structural proteins. The only way you're going to make antibodies to structural proteins is you become infected, right? So if you have antibodies to like the N protein or one of the other structural proteins, we know with certainty that you've been infected by SARS-CoV-2. If you only have antibodies to spike, we know that you've only been vaccinated. So one of the things that we could do would be to have a kind of a twofold test. One to look for spike proteins and one to look for the other the structural protein antibodies. And depending on which profile we see, we either know that this person has been infected by the virus or not, or have only been vaccinated. But what I really want to see is a test that will tell us whether or not a person is protected. And do they need to be boosted? Because if we had that, Dr. Sherrod, we would need to be boosting people every 4 or 5 months. Yes. I have no intention, I'm just letting you know, I have no intention of getting a shot every 3 or 4 months. Okay, if I have to wear a mask and social distance and whatever I need to do to protect myself, I'm going to do that. I'm not getting a shot.
And the last question I have is where, and I know this is a hypothesis, but where are we going? Where do you see us going with this pandemic, endemic, whatever you want to call it? Where are we going?
I'm keeping my fingers crossed. There are 3 high-profile groups, maybe more than that. These are 3 that I know of, that have some pretty compelling evidence. They've identified a way to make a vaccine that will elicit antibodies that would neutralize all the variants. If that happens, and we got vaccinated with those vaccines and boosted with them, that would mean that, like measles, perhaps, maybe not like measles. Measles is lifetime protection. But maybe like some of the other vaccines we have that give you protection for years, we could achieve that. Okay. But the problem with this is measles does not mutate like SARS-CoV. Flu viruses are segmented genomes, so it's all a guessing game. I shouldn't say that, but to some extent it is. You're trying to predict which of the segments of the genome will be put together, and you're trying to base your vaccine on which segments will be put together. And so we have the efficacy of 40% some years to 60 or 70% some other years. But we're hoping that with SARS-CoV-2, there might be some predictive convergent evolution, and there'll be dominant epitopes. We'll make vaccines those epitopes, and we'll call it a day. And that's what my colleagues are trying to do, is to identify epitopes that are dominant and convergent, make vaccines based on those. We get vaccinated with those, maybe once every 2 or 3 years, we'd have to get that vaccine. That'd be a much better picture. That's what I think is going to happen, Dr. Schwab.
Is there anybody else with their hands up that we need to recognize? Dr. Neighbor Stevens, Dr. Wonks, Dr.
No other hands up, Dr. Maxey. There was a question to clarify something, Dr. Hildreth. I believe you said that as you went from Moderna for the first 2 to Pfizer for the 3rd one, that was it Novavax you said was your 4th shot?
Yes.
Okay, great. Thank you.
Novavax
That was the question.
Novavax is the only vaccine that's approved in the United States that's protein-based. All the others are either adenovirus or mRNA.
Yeah.
So Dr. Maxey, there are a couple of points in the chat, and a couple of these just came to me directly. I don't know why, but folks who have read some of the things that we hear from time to time, that this person had the vaccine and then got Bell's palsy. This person had the vaccine and then had a myocardial infarction or heart attack. And so I think is it fair, Dr. Hildreth, for us to say this afternoon that you and your colleagues you mentioned earlier are paying attention to those things?
Absolutely.
And if there's some trend of worry that is a current concern for you, you would have shared that with us. Is that fair to say?
Absolutely. Because I've been putting my reputation on the line by talking about the safety of these vaccines. I've insisted that my son, my daughter, my wife, my sister, my brothers, all my relatives have gotten vaccinated based on my asking them to, some reluctantly, but they thank me now because, one got COVID and survived it. We had other people in my family who didn't take the advice, who got it and are not with us anymore. So if there's any inkling whatsoever that these vaccines are going to be a problem, believe me, I'm going to be the first person to. Okay, I think that's the least I can do given all the advocacy I've been doing on the part of people getting the vaccines. But I can tell you that being on the VRPAC gave me a chance to see all of the data for myself, right? That's one of the advantages of being on the FDA panel. If we ask for some data and it's not provided to us, that would be known. If we see some data that's disturbing to us, that would also be known. And for example, there was a drug that was being developed by one of the drug companies for COVID-19. Its mechanism of action was something called catastrophic mutation. It actually drives mutation of the virus, okay? And the theory is that it would induce so many mutations in the virus that the virus could not replicate. The problem is that for those persons, and you know this, physicians on the call will know this, some patients do not complete their course of treatment. And for antibiotics, that's bad news because it simply promotes mutations and bacterial mutations, and you get drug resistance. So imagine having a drug out there being taken by millions of people that actually causes the virus to mutate. You have much more chance of deriving variants of concern than not. So I voted no along with 12 of my colleagues, but 13 of my colleagues thought it should be approved, okay? But I made no secret of the fact of what a bad idea I thought that was. And I got into trouble for saying it, but hey, you shouldn't have asked me to be on the panel if you didn't want me to express my opinion. So
Amen.
all I'm trying to say is that when I have a concern about something, I'm not bashful to say so. And I've actually shared with the CDC director how poorly I think they're communicating about some of the decisions they're making. And so, but you know, people listen to what they have to say, but I do think they should do a better job of communicating the rationale for why they're making recommendations so people can make their own decisions. People are smart. Right?
Dr. Hildreth, I'm gonna make sure you address the question on long haul, but that first one, is Dr. Oliver Brooks on, Dr. Wilkes? You see Dr. Oliver Brooks, he's a member of the ACIP with you. But would you kindly address long haul syndrome and what we need to worry about with that? What percentage of us that have had COVID, and I had about a month or so ago, and I was so glad I'd been vaccinated. But what percentage of us that have had COVID or get COVID can expect to have symptoms of post-acute syndrome?
So Dr. Maxey, depending on which study you look at, the range appears to be somewhere between 10% and as many as a fourth or 25% of people who had COVID will have post-acute sequelae of COVID-19 or past. And one of the things that's become really clear over the last several months is that the lung is the primary site of infection, but there are many other tissues in the virus in the body that gets infected. And one of the most intriguing ones that I'm aware of is a study to show that adipose tissue, adipose cells, can be directly infected by SARS-CoV-2. And we thought that obesity was a risk factor because when you're obese, you can have problems with your respiratory system, and those problems would lead to, being more acute disease for SARS-CoV-2. But now there appears to be the case that the adipose tissue gets infected, elicits inflammation that contributes to the pathology that we're seeing. So, and the thing that's intriguing about that is the adipose cells do not express ACE2 at high levels. So it looks like the virus is going into those cells by another mechanism. There's evidence that cells in the kidney might be harboring the virus. My point is, since the receptors all over the body, that long list of symptoms that people get with long COVID might be explained by residual virus lingering in our tissues in places other than our lungs. The confusion, the lethargy, some people are having some cardiovascular challenges. We think some of these things may in fact be related to the fact that the virus has not been completely cleared from our system. And that is why I've worked so hard myself not to get COVID. I'm more worried about long COVID than I am about COVID-19, to be honest with you. From the SARS-CoV epidemic that happened in 2003, there are people who couldn't go back to work for as long as 2 years because of the lingering challenges they had with this. The other problem I'm worried about, and we talked about this on President Biden's Health Equity Task Force, if you present with a syndrome that cannot be measured with an objective test, if you are a black or brown person, or if you're a woman in some cases, you may not be taken seriously, right?
Mm-hmm.
One of my colleagues, who is a young, amazing assistant professor at an institution in Texas, got COVID, then got long COVID, went to the emergency room, and they swore that she was using drugs. So they did a complete workup for drug use and did not accept the fact from her, A, that she had never used drugs in her life. She actually showed them that she's a professor at this, university in town. But they refused to accept that and insisted she was a drug user. I told her she should sue them, but she said, no, I'm not going to do that. But my point is, one of my most major concerns is us, when we get long COVID, may not get the same reception when we present to physicians as we might otherwise get, because you can't necessarily diagnose this yet with a definitive test you can do that provides objective measures. Right. So Dr. Maxey, one thing that we're insisting on, and we've actually said this to the president himself, we got to make sure that health systems throughout the country who have patient panels that don't include us must be obligated to deal with long COVID. Because if they don't do this, we're going to have a major problem in our hands. Not to mention the behavioral health implications of this, which are going to be huge. So I don't have an answer to your exact question, but it's a really important question because many people that we know are going to get this and they're going to need help.
Well, Dr. Maxey, Dr. Clark raised her hand. We could unmute Dr. Clark.
Can we get her?
I know we're getting close to time, Dr. Maxey, but
yes. Is
it possible to get Dr. Clark unmuted? Dr. Michelle Clark?
Got it.
Good. There we go.
Thank you. Thank you. And my phone's ringing. Hold on a second, Nick. I just wanted to say that for your colleague who went through that horrific experience, if she doesn't want to sue, she should at least write to the hospital administrator and perhaps do an article in the local news. That's all. Thank you.
I'm going to pass that along.
Amen.
That's a great suggestion.
Dr.
How you doing?
Dr. Maxey?
Yes, sir.
I know we're running close to time, but
let me turn off this.
Since Dr. Neighbor Stevens got to make her pediatrician comment, I'd like to make a psychiatrist comment. I thought Dr. Clark was going to maybe talk about this. There's something in the chart that in the chat that I think is worth us taking a look at. And I just want to read this first part of it. How can anyone trust a messenger when the messengers like Fauci are getting paid directly or indirectly from the pharmaceutical industry? This includes the CDC, the FDA, the NIH, including colleges, universities, medical schools, hospitals, etc. And the reason I bring this up is folks like me, I'm a physician, I tend to dismiss stuff like that. Oh, you just won't. But there are people who I think struggle with this. And I think Dr. Hildreth, maybe you've experienced this before. I don't know who this person would listen to. It is scary when I think about the fact that if you're not listening to any of the doctors, any of the scientists, and another part of this comment is that, have you looked at how many people are being killed by the vaccine? Well, I think it's important for us when we see comments like this to engage with these folks, at least to say, my goodness, I'm concerned because there's no one you're listening to except someone who's getting paid by how many views they get or how many likes they get. They're getting paid too. So maybe you've experienced this, Dr. Hildreth, and I thought we should take it at least seriously because there are folks who want to dismiss everything.
Sure.
That's a great question.
So over the last 2.5 years, I've had my share of people who made those same accusations for me. I could not serve on the FDA panel if I were being directly compensated by any of the drug companies, not the ones that make the vaccine, any of the drug companies. I had to fill out this 20-page financial disclosure before they let me be on the panel. And I thought it was intrusive, obtrusive. I mean, I was offended by it, but I understand why it is. So I could actually say that I have no benefit whatsoever in recommending approval of anything, because it's not going to benefit me or anybody in my family. And that's true for every member of that panel. And if you listen to the FDA meetings, you'll actually see that they call me out specifically because Meharry Medical College did part of the Novavax COVID-19 vaccine trial. I'm not in it. I'm not the PI of the trial, but I'm president of Meharry Medical College. Even the fact that I'm president of an institution that was part of a trial means that that has to be disclosed for people to know it. I'm not benefiting from it. I'm not in the trial, but I'm leading an organization that was part of the study for Novavax. Dr. Fauci has to do a financial disclosure each and every year to reveal who he gets paid by, where his income comes from. And I promise you, there's no drug company listed because he wouldn't be allowed to serve in the role he's in if that were the case. So I don't know where these ideas come from. I know people get more likes and more followers by saying these things, but none of them are factual, okay? I can just tell you that. And on my part, when I sat on the FDA panel, when we reviewed the vaccine for children from 6 months to 5 years of age, a sitting congressman from Georgia got on the public comment portion of the meeting, and his almost exact words were, he's a Republican, when we take over Congress again, we're going to pass legislation to hold all of you, referring to us scientists, responsible for killing children. Okay. Can you imagine being threatened by an elected official for sitting and offering your scientific knowledge and advice to the FDA? I'm not killing children. If I thought I was killing children, I'd be running away from the FDA. Okay. My point is, some of this stuff is so out there that you wonder, is it even worth it? Because there have been a few times when I thought, well, maybe I should just keep my mouth shut. But if I were not sitting on that panel, and there were no folks on the panel who looked like myself and Ovita, what would the conversation be then, right? So I don't know what to say, Dr. Walls, to those kinds of things, but I can tell you most of it is completely false, misguided, and those individuals do not either understand how the process works, or they're just perpetuating this nonsense to get more likes and more followers. That's all I can tell you.
Well,
Thank you, Dr. Hildreth.
Thank you for having joined us today. And I'd like to recognize you for final comments, and I don't know if you're comfortable with this at all, but there has been a lot of data, information, misinformation from Joseph Kennedy and others that I consider misinformation. And some people have even said, and I'm one of them, that there's a lot of this misinformation aimed at preventing our community from using these vaccines and protective measures. Currently, I'd love you to comment on that if you would. I won't insist on it, but I invite you for final comments before we get our comments from our moderators left.
So, Dr. Maxey, I would just actually have people understand what science is and what science is not. Science is not knowledge or even factual, to be honest with you. Science is the process by which we pose a question and try to find an answer. What that means is, as our tools get better, the answers we derive with those tools get better. And our knowledge can be more useful to people. And we had never experienced anything like this before. We've had pandemics before, but never a pandemic like this. So the virus is new. We had a president in place who did not take it seriously, who insisted that politics supplants science, right? We had people in our own community who spoke about the past atrocities, and they have a right to do so. As I said, I understand why people are so passionate about this. when I read about it and still think about what happened to those 600 Black men in Tuskegee and how they were treated and all, it makes me very angry. When I think about Black and brown women who've been involuntarily sterilized, that makes me angry. What makes me even more angry are those people who want to kill us by having us dismiss the power and utility of science. I've been studying viruses for 40 years. I think I know a little bit about them. And if I were to keep my mouth shut in a time like this, my mother would disown me. My God would probably turn his back on me. There's this story about, I think her name is Naomi. And the catch line is, for such a time as this. So Dr. Maxey, I believe that everything I've done in my life maybe prepared me for such a time as this. And if I kept my mouth shut, despite the people who had such horrible things to say about me and what I was doing, that I don't know, it would have been a unforgivable thing from a lot of points of view. So I'm just saying, I'm just a scientist who cares about his people, trying to make sure we can protect ourselves as best we can. I mean, science is not perfect, but we take science for granted every day. Every time you sit in that aluminum tube 6 miles in the air, you're trusting science. Every time you go to the grocery store and buy food that's been preserved to make it last long on those shelves, you're trusting science. Every time you go to a physician and he recommends a drug you take, you're trusting science, because science is what let us know that it would work and not harm you. So for us to accept science every day in our lives, but to all turn our back on it at a time like this doesn't make any sense to me. And I'm going to leave it at that, Dr. Maxey.
Well, thank you so much. Dr. Jocelyn Hines, final comments?
Dr. Hines had to drop off. She is at a medical meeting this afternoon and joined us for as long as she could, but left a very positive comment for Dr. Hildreth.
Okay, well, Dr. Walsh, it's just you and me.
Okay, so Dr. Maxey, does that mean I can take twice as long? No, I didn't.
No.
So Dr. Hildreth, thank you so much. I appreciate you taking the time to address some of the concerns about misinformation, taking time to put your personal credibility out there, because I think it matters to people when we get personal and when we talk about how things matter to us. So I really appreciate you doing that. There is so much misinformation that is out there. And I think one of the things that's different today than maybe with previous pandemics is that people like to have their own facts now. I am going to decide what's true. I'm not going to listen to you and your 40 years tell me what you know about viruses, because I just read something and now I know more about viruses. And because we live in a world like this, it's important for us, when we care about our people, to be willing to speak up. There was a comment in the chat, Dr. Hildreth, for you that reads, "Please don't keep your mouth shut. We need people to continue to speak up against the misinformation." And I think sometimes when we are out there, it's important for us to hear those comments. It's important for us to know that there are people who appreciate the fact that we are speaking out. My last part of this comment, Dr. Maxey, is that the Black Health Trust is a place where we speak about truth that is verified to the extent that we can verify it to a scientific level of certainty. That's what we do on Sunday afternoons. So guests like Dr. Hildreth allow us to continue to say that we do that. So the last thing is this: these programs are available for folks who aren't able to be here on Sunday afternoon. Dr. Maxey mentioned that we stream live on YouTube and Facebook, but it's available after the fact as well on those platforms. So I would encourage folks to share this program and other programs with folks who weren't able to make it this afternoon, because it's good information, man. It just is. Thank you so much, Dr. Hildreth.
Dr. Hildreth, again, thank you. I didn't go to church today, but your words gave me almost salvation. And we thank you so much. And you've been an honored guest. Thank everybody for joining us on this Sunday afternoon. Enjoy the rest of your day. Have a good day, everyone.
Thank
Thank
you so
you.
much.
Good day, everyone.
Appreciate the opportunity.
And thank you, Simon Cooper and Skylar. Thank you, Doctor.